these are some issues and some thoughts about that. What a lot of the activity at ASH was about transplanting CAR T. And one of the messages, as everyone talks so much about CAR T, and I'll talk about CAR T, but increasingly, it's just important to recognize that transplant and allogeneic transplant remains critically important. It still cures people. It's got a long history, and it's not going away anything soon. As a matter of fact, there's more people getting allogeneic transplants now than ever before. And again, the important message here is it's no longer your last resort. It's not, and we know that for a long time. Sometimes we see patients and right off it's transplant has to be what's given. And this is the usual IBMTR slides that show that um, allogeneic transplant is still going up. This is as of 23. Autologous transplant has leveled off, and not surprising, CAR T is going up, and a lot of this business is moving over here. And we'll see that change over time, and we're going to be interested to follow that. So donor age does matter. And there's a couple of published studies recently looking at this issue of donor age in transplant. And because what we're finding is we are transplanting older and older patients than we ever did before. And that's for older recipients, but I'm sorry, this is the donor age for the donor, because now we know that younger donors are giving people a improved survival compared to using older brothers and older brothers or sisters. So we have used to take HLA matched siblings up to age 65, 66, 67, close to 70. Now we are moved to taking unrelated donors that are young over a matched unrelated young donor over a matched family member. And so this is the edict that comes out of these studies that young donors under age 50, there is now data to say you have overall survival is going to be improved and disease-free survival is going to be improved for myeloid malignancies. Why? Well, there does seem to be younger donors have lower risk of relapse and improved disease-free survival. And interestingly, what we actually published a couple of years ago, and increasingly people are seeing older donors are more likely to have CHIP, clonal hematopoiesis of indeterminate potential. And an older donor that has CHIP before you even start and you do the transplant, the risk of graft versus host goes up. And it makes sense because we know that CHIP often makes you more inflammatory. CHIP actually is associated with higher rates of coronary artery disease in the same way that your cardiac vessels are more inflamed. And these older donors are more likely to have CHIP. And so younger donors will not have that. And so this is an example, though, importantly, though, most of the data about younger donors versus older donors is done in the old with a lot of long-term data. You can see here's retrospective study between 2010 and 2020. Well, that's the time period that everybody still was getting calcineurin inhibitors with methotrexate. And so this study just got published, and this was presented and talked about at ASH. If you look at donor type, and here's calcineurin inhibitors with a mismatched unrelated versus a mismatched, with getting calcineurin inhibitor and methotrexate. I'm sorry, a match. So mismatched unrelated versus matched. You can see donor age at 20, even a mismatched versus a matched in the setting of calcineurin inhibitor and methotrexate, not that much of a risk. But if you start to go up to donor age of 50, your risk of complications goes up dramatically. Interestingly, whether you have a mismatched unrelated or a matched unrelated with post-transplant cyclophosphamide, that seems to be neutralized. I'll skip that part. So that was one piece. And then what we're showing is now with data, this was published by Brian Schaefer, and there's now, and I skipped these, but there's now the National Marrow Donor Program in the U.S. has been running two new trials called Access and Accelerate, which are testing can they maximize and improve upon the dose schedule of post-transplant Cytoxan. Everybody gets tacrolimus and MMF, but the post-transplant Cytoxan, should it be 50 milligrams per kilogram times two? Should it be a lower dose? Could it be a different dose schedule? Those studies are going on. But importantly, if you look at overall survival and GVHD free, relapse free survival, whether you have a 7 of 8 or 8 of 8 in the setting of post transplant cyclophosphamide, there really is no statistically significant difference. Calcineurin inhibitors, maybe there's a reduction of overall survival with the 7 of 8 because of more toxicity. So we seem to neutralize that. And this is why is it neutralized? Well, you see that if you look at acute graft versus host disease or moderate to severe chronic graft versus host disease, and if we just look at the 7 of 8s, here's a 7 of 8 with PT-Cy versus an 8 of 8. But with calcineurin inhibitors, if you have a 7 of 8, you've got a 50% risk of grade 2 to 4 graft versus host disease. And the same thing with you look at chronic graft versus host disease. If you look at PT-Cy, with whether it's a match, full 8 of 8 or a 7 of 8, it's much lower than if you do calcineurin inhibitor-based GVHD prophylaxis. So we're now saying that we are now using younger donors and we're using post-transplant cyclophosphamide. This has now become the standard. I know you've been using this for years, but this is now officially taking over in the United States. It started with Haplo from the Johns Hopkins experience, but now most unrelated and most match-related transplants are now using post-transplant Cy and calcium, cyclophosphamide. I'm sorry, cyclosporin or tacrolimus and methotrexate is pretty much disappearing in the United States. And there are some new research trials coming in. I'll talk about ORCA in a minute, but these are other variations that you can use that are still being tested. Now, where do we come? What happened in the United States? Well, also, another big trend is myeloablative transplant. Big Bu flu, big Bu Cy, Cy TBI is being diminished. And a lot of it has to do with the fact that we're treating older and older patients. And as we treat older and older patients, they can't tolerate myeloablative. And there was some data recently presented that 70% of all myeloablative transplants in the United States now is reduced intensity myeloablative, basically mostly all fludarabine and melphalan. And this is based on the paper. This is BMT CTN 1703, which compared the post-transplant cyclophosphamide with tacrolimus methotrexate. And what it showed is, and it's a primary endpoint of acute, at the end of 1 year, graft versus host free, relapse free survival, or GRIFFIS, was far superior for patients who got the post transplant cyclophosphamide versus the patients who got the standard classical calcineurin inhibitor and methotrexate. And it does seem that all of that was related to graft versus host disease. And so relapse was actually relatively the same. And so interestingly, at the end of 1 year, overall survival still was the same, but the GRIFFIS was what was impacted. But importantly, we did not see higher risk rates of relapse. Back in 2006, there was a BMT-CTN trial of haplo identical transplant using the full dose PT-Cy, and it was often for advanced leukemia. And there was a 50 and with reduced intensity. Most of that was a lower intensity regimen than the fludarabine melphalan, and that had a 50% relapse rate at 1 year. What we see now, with where we've evolved, we now have a relapse rate of still 20% at 1 year. So superior data. Now, what's interesting is that, and this is on the fast track to FDA approval. This is work from Rob Negrin. Dr. Negrin is now the president Ash. He took over in December, but in his laboratory at Stanford for years, he was looking at T regulatory cells, and he came upon it. He did all the laboratory animal experiments, and it led to this basically equation that everybody could have a peripheral blood stem cell transplant. You do myeloablative conditioning, but what you do is on your donor, peripheral blood stem cell product, you sort the cells. You purify CD34 positive stem cells, and then in the eluate, which has all the T cells, you take the eluate and you sort for Tregs as a CD20, CD4 positive, CD25 high population, and then you have T cons or conventional T cells. And so basically it's a single graft from an individual peripheral blood stem cell product from a single patient, but you purify 34 cells, T regulatory cells, and the residual conventional cells given in a specific prescribed dose. And then on 5 days, starting afterward, use tacrolimus as a single agent, no other drug. Well, these data have just been published in December. In a randomized trial. They were first presented last year at EBMT, but they compared it to standard transplant. Again, tacrolimus methotrexate, not post-transplant cyclophosphamide. But what they showed is these patients with this graft-engineered product have a improved overall survival with a hazard ratio of 0.5. Definitely a GVHD-free, relapse-free survival, and it was all based on chronic GVHD reduction, minimal chronic GVHD in this setting. And if you look at relapse, the relapse was almost, at least for deaths due to relapse, was equivalent. So with the graft engineered product, there was not an increased risk of relapse, but there was a reduction of GVHD and overall clinical improvement. They are planning they are planning a prospective assessment against post-transplant cyclophosphamide, but all they could do now is do a IBMTR collaboration or connection to show that the ORCA product, this graft-engineered product, compared to standard PT-Cy, that 1 year relapse mortality, non-relapse mortality was 3% with the engineered product, 16% PT-Cy, and then also you could see overall survival was improved at 94% versus 77%. So the FDA, they have filed for FDA approval in the United States. The approval date is going to be in April 2026. Now, will they be accepted? Well, who knows? But let's pretend they are. What we don't know is what are they going to charge. Now, we know that Gamida which had a graft engineered cord blood product which was FDA approved based on faster neutrophil engraftment and lower infection rate, not an overall survival benefit. They're hardly being used at all because they put a price tag, honestly, of $325,000 US. I don't know what these guys are going to price, but when you look at this survival advantage, you're going to ask the question of 94 versus 77 at 1 year. Is that 10% increase or 15% increase in survival going to be worth potentially $300,000-$400,000 a year per patient more? That's a question. I'm just saying I don't know. I'm making no projections. I have no insider information. I'm just looking at what they're not looking for it. Why am I basing this? Is they're going for their FDA approval as a cell therapy and not as a transplant product. As a cell therapy, you're matched up against all the drugs, which are typically in the $400,000 to $500,000 range. So speaking of cell therapy, classical cell therapy now, or the non-classical transplant, again, if we talk about CAR T, just to remind, we have all these diseases that are currently approved for by the FDA, ranging for acute lymphocytic leukemia was first, and most recently was chronic lymphocytic leukemia. I would point out that marginal zone lymphoma also was recently approved, but these are all, targets, including multiple myeloma, and it's based on two target antigens, CD19 or BCMA. These are the current commercial T cell products, the CAR T products in the United States. There's 7 of them. And there's 2 FDA approved T cell therapies also. And I know I've said this before. This is afami-cel is for synovial cell sarcoma, and lifileucel is TILs for melanoma. But these 7 products are all commercially available. I put this in for fun. At the end of every month, you can go to the FDA and ask, and there's on their website for the CBER, they will list all the updated products. This is the current list of products approved by the FDA for cell and gene therapy. And these are going up by 8 to 10 per year. And so we know that the most expensive is about 3.5 million. The least expensive, I think, is around 100,000. But it's big money in this business, but it's a lot of products. There's a lot has been done. There's certainly the cord blood is listed for cord blood banks, but also drugs for orthopedic repair, um, the oncolytic viruses, as well as genetic disorders are all incorporated. If we look at CAR Ts, we know again this is IBMTR data. The IBMTR shows in the United States the number of procedures every year are going up. There is a major projection. This is from this one group that looks at marketing, that CAR T by over 10 years is going to go up. The value of the market is going to go up, fivefold to an $80 billion industry. Is that right? 1.5 million. Yeah, no, an $8 billion industry. But either way, when you look at it statistically, CAR T recently, there's data to say it's up to 900 clinical trials, and there's over 1,400 different cell therapy pipelines globally. And that includes some preclinical work that haven't yet gone to clinical. Of the patients treated, there was over 35,000 patients treated with the FDA products. There's 10,000 patients treated in 2024 alone. This was interesting. EBMT, there's data from EBMT. This just came out earlier in January that they now think worldwide there's been over 100,000 CAR T procedures have happened. And this includes a lot of activity in China where they've been doing, CAR T in multiple indications for quite some time. This is our own cell activity. We've officially did hit to 150 procedures in 2025. And this is 2018 is when we first did our first commercial products. And so to start with, let's just look at a couple of the CAR T indications. I want to talk about multiple myeloma. And one of the questions, is there anything better out there? So these are the 2 FDA approved products, idacaptogene and ciltacabtagene. They were first approved for 4th line and beyond. Now they're approved for 2nd and 3rd line. These are the data that led to the early approval. And so here is idacaptogene at a median time to progression of like 1 year versus a median time to progression of 5 months versus best standard of care. For ciltacabtagene, the median time to progression for standard care was 1 year, and the median time of progression was not reached for ciltacabtagene. This is what led to approval. I point out these are not comparable studies. Everybody on the study on the left had to have failed CD38 antibody. They all had to be triple refractory. On the right here, this includes patients who were only listed as lenalidomide failures. So patients had their induction therapy, had their auto transplant, they were on lenalidomide, and on lenalidomide refractory status, they could move right to CAR T. And so a lot of these patients have never seen daratumumab or CD38 antibody, so they are a better risk group. So you can't compare, but you can see compared to the best available standard of care, both of them were superior. Now, what we know has gotten a lot of people excited. I think I presented this in a talk on myeloma last October. Is CARTITUDE-1, which is the original siltecabtagene study, um, showing that was in for fourth line, and many of the patients were 7 and 8 lines of therapy before they got to CAR T. Well, we know that in CARTITUDE-1, the median progression-free survival was about 2.5 years, and that's been known for quite some time. What was published by Jagannath from New York City in 2000 in the spring of 2025, it was May of 2025, is they now know that 1/3 of the patients at 5 years are still without progression from that single dose of CAR T, with an overall survival of a fourth line, fifth line, sixth line patient at 50 something percent. So this has really got a lot of people excited, especially right here. If you look at 5 years, there's again, it's still between 5 years and 6 years and a half, short time limit, but no late relapses. They're starting to see a plateau. And so people are wondering whether CAR T can cure myeloma. Um, I'll skip that for now. I think this is next up. Again, is there a better product? Well, this is idacaptogene, single SCFE. This is the siltecaptagene, which has got this really bivalent double binding SCFE. And there's a thing that's called anitacaptogene autoleucel. This has got a small binding site. It's a graft engineered, genetically created CAR T, where it's only taken basically the anti-idiotype region of the entire fab and constructing it to bind to the T cell machinery. One of the advantages, it's so small that when you go to use a lentiviral transduction of a population, you get many, many more copies into the target cell than you do when you use the whole large molecule. And getting more copies means you have higher expression rate, and you end up needing less cells to get the target number of cells that will deliver a signal. Interestingly, also, it's got this, because you don't have this really large construct with its steric effect, it binds fast, it delivers a signal, and gets off quickly. So it's an easy on, easy off um, molecule. So you don't get tonic signaling to the CAR T, and so you have basically, they think, a better product. Well, there was a phase 1 trial that showed that the median progression-free survival was 2.5 years. That looks pretty good. That looks exactly like siltecabtagene. Um, Median overall survival not reached. Again, that looks like siltecabtagene. The safety profile, there was no late neurotoxicity, which looks better than siltecabtagene. And now there's a phase 2 trial, which is an expanded access trial to get up to 100 something patients. Now we were part of this. This is your classic, you know, um, non-randomized, single-arm trial, lymphodepleting chemotherapy. You manufacture the product, you give the cells, and you wait with long-term. And there's a target of 1 × 10⁸ CAR cells. Well, you see here, 96% overall response rate, 74% complete remission rate, and time to first response was 1 month. Importantly, the median time to becoming MRD negative at 1 to 10 to the minus sixth was 1 month. And overall, 80% of patients got to that really deep clonoSEQ sensitive level. Importantly, in this is Imagine-1. Again, this is the phase 2 study. The progression-free survival at 2 years was 60%. Overall survival, 83%. Interestingly, CRS grade 1, very low grade 2, no grade 3 to 5. And when you look at ICANS, there was a total of 8 patients out of like 115 or so with ICANS at all. Very low risk. So no long-term neurologic toxicities and very low CRS, very low ICANS, and what looks like an equivalent outcome as Cilta-cel. So everybody's excited about this product. Again, we were part of the non the phase 2 non-randomized. There's now a randomized trial against standard of care. We're part of that. It's expected, and it's out there now that the the FDA basic license agreement um, is expected to be filed in February or March. That there will be action potentially by September, by July, sorry, by August or September. This drug may become available. This may be the next CAR T. Now, what's very new, this I found the most exciting moment of CAR T the most exciting thing I saw at the ASH meeting. And so these are neighbors of yours in Melbourne, Australia. But there's a three center collaborative study in Australia with a company called Colonia. Now, this is your standard how we make CAR T cells, identify the T cells, engineer them in the laboratory, expand them, send them back to us, and we infuse them and wait. And there always has to be lymphodepleting chemotherapy. Well, this is an in vivo CAR T product. So this is going to be the first clinical presentation of this particular product in humans. And what they do is they take the particles, no lymphodepleting chemotherapy. They have a set dose of particles. You just inject them, and then you wait and watch and see what happens. And so, what is the product? It's very interesting. This is the the vesicular stomatitis virus, or VSV. This is in the the rhabdovirus family. That's what rabies is in that family, the bullet-shaped particles. Well, there's a thing called a fusogen, and the fusogen makes it stick to a target antigen. Typically, it binds to the liver very aggressively, but what they've done is they mutated it so it will not bind to liver. And I took all those slides out. But what they did is they also expressed on the capsule wall a CD3 CAR. So what it will do is this is the capsule of the VSV. This is the Trojan horse because inside the capsule, they got a lentivirus vector with the classic BCMA CAR with a 41BB target. So a lentivirus hiding inside a VSV capsule. They inject that directly into humans. And what they said is the plan was a 3x3 dose escalation, dose level 1, dose level 2, dose level 3. This is going to be dose level -1 if you ran into toxicity at dose level 1. And then if this would, based on what they got, there'd be an expansion phase to identify the recommended phase 2 dose. So that's the study. And it has to be 3 or more lines of therapy and 3 sites in Australia. Well, this is what's remarkable. The first 3 patients, first of all 4 patients, very high risk myeloma. 17P, 3 of them, 1414, all of them with multiple lines of therapy. A number of plasma cells, one had 60% plasma cells, all were triple refractory. Now, here's what happened on the first 3 patients, dose level 1. All 3 patients at the end of 1 month were MRD negative to a level of 10 to the -6th. They followed them out up to 6 months. The first patient's only at 6 months. This is very early, but already in a stringent, complete remission. In all aspects have cleared their free light chains. M spike comes down, soluble BCMA down. So what they did, based on everybody responding at dose level 1, the backup in case of toxicity, they went and treated a patient at dose level -1. And what they found is that patient also at the end of 1 month was MRD negative at 1 in 100,000. So it is incredibly sensitive testing. It seems to be an amazingly potent product. What will happen? Who knows? But this was the chosen to be the #1 abstract for the late breaking abstracts, important enough for us all to see. So also this is just says the cells expanded, very similar without lymphodepleting therapy. All patients expanded, and it looked at like and they actually look like normal CAR T expansion. So everyone's excited. Everyone wants to like say, hey, can I borrow, get in on the study? Who knows? So I was going to talk a little bit next, if I can. It's 5:40. I think I can get about 5 or 10 more minutes. Um, what about lymphoma? Well, lymphoma is always there. Lymphoma is probably the biggest reason why people get to CAR, although myeloma will probably replace that in the future. Um, what about toxicity? Well, we know that there's all this just tells us what happens when you get, systemic inflammatory response. These are all the potential organs and all the different symptoms and signs associated with all those organs. And this slide is Jim Korkendorfer's, he's the one who helped develop the originally axicabtagene. And we know that each product has their own specific toxicity and their own specific, you know, likelihood of CRS. And so this is in orange is the overall rate of ICANS, in blue is the overall CRS, and also how many with advanced CRS was in green. And we know that um, Axicell tends to have the highest rate of ICANS, as does Brexicell. These are both CD28 CAR Ts. These are the 41BBs CAR Ts right here, and they seem to be lower. So this was an interesting presentation that from University of Minnesota, from Veronica Bacanova. This is using the cell. There's an 8-center CAR T consortium. Always just use one of the centers. But I thought this was an interesting study that they did. It's a composite of a shared database of 630 patients, and it's like the first 6 years, and it's all looking at lymphoma. And in the same way, so we always look at CRS independently. We look at ICANS independently. We look at relapse independently. We look at overall survival. Well, what she did is saying, well, why don't we do what we do with what we're doing now with transplant? Is we have Griffiths, GVHD relapse free survival. What happens if we have CAR T toxicity free relapse free survival? So is that going to be a new endpoint that may be of interest? And out of this, you could see that over 50% of the patients were axicabtagene, a third were Ticicel, and then only about 10% were lysocabtagene. These are standard endpoints. This is what you expect. Overall response rates to be these are the reported response rates. Lysocell got they actually have higher response rates, but of course, Lysocell by the time they showed up on the market, we had better drugs. We had more than chemotherapy. We had tafasitamab, polatuzumab. We had better drugs to get somebody to CAR T. But either way, CAR T had better responses. Progression-free survival by product, statistically significant. Lysocell looked better, and CRS. This is what we think of today, and this is what we saw in the data from this consortium. So, Ticicel and Lysocell equivalent for CRS. Axicell higher, and higher rates of ICANS with Axicell than with Ticicel, Lysocell. But probably Axicell may be superior to Ticicel for progression-free survival. So the question is, what happens if you look at toxicity-free survival versus toxicity, complete remission with toxicity? So, and toxicity is defined as CRS grade 3 or more, or ICANS grade 3 or more. So advanced CRS, advanced ICANS. So if you look at now 2-year overall survival, someone who is toxicity free, clinical remission, relapse free, now has an 86% chance of being alive at 2 years. And we know being free of disease at 2 years predicts the 5-year cure. If, on the other hand, overall survival, you could be relapse. This is going to be complete remission with toxicity. These patients haven't relapsed. Again, not no relapse at 2 years predicts 5 year survival, but we know we've got a 20% less survival, and that's really often occurs at 9 to 12 months. And this is probably infection related to the prolonged complications of dexamethasone and the prolonged complications of high grade CRS or ICANS. Of course, if you were a non responder, your likelihood of being alive at 2 years is also low just because your disease is growing. This is after CAR. So patients do progress. And again, for progression free survival, you can see that even then, a complete remission with toxicity versus toxicity free, there's a trend also statistically significant benefit. So it's an interesting endpoint. Um, and we know it has nothing to do with relapse. It's completely all based on non-relapse mortality, that the toxicity free survival versus, those with toxicity, you're losing people. And so we have to basically, this proves without a doubt that CRS and ICANS does lead to mortality that has to be addressed in the future. I'll skip this sort of stuff and saying yes, the conclusion, the composite endpoint may be proved to be as useful in the future. And non-relapse mortality is fivefold higher in the patients with a CR with toxicity than those without. And what about ICANS? Well, I'm going to skip this, looking at the time, because this is work that came out of, we presented, which is that how do we treat ICANS? And we think we've got the way to treat ICANS, which will be high dose anakinra. I'll do that at a different lecture. But final thoughts about lymphoma is just to point out follicular lymphoma, and that not to ignore follicular. Follicular tends to be ignored, by the way, for CAR T. There's so many things to do for CAR T. I can do bispecific, I can do bend, I can do whatever. I can do loncastuximab. Well, this was a publication that looked at axicabtagene versus Scholar 5, which is follicular lymphoma, relapse refractory follicular lymphoma. They looked at third line and fourth line. This over here are pictures of fourth line. And you could see that, again, this is retrospective comparison for overall survival, but certainly for progression-free survival. CAR T was superior to other standard of care, other treatments. Well, what do we know about CAR T that's now published? Well, this was published in the late fall of 2025 in the JCO. This is Zuma 5. This is the official 5 year end of study data of almost 160 patients treated with follicular lymphoma or marginal zone lymphoma, median follow-up over 5 years. And what we have is a median progression-free survival of over 5 years after CAR T, again, with a suggestion of a plateau showing up. Now, what was presented at ASH was the 5 year data now of tisagenlecleucel for relapse refractory follicular lymphoma. Basically, similar data, very high risk disease, POD 624, bulky disease, high metabolic burden. But again, overall response rate 86%, CR 70%, median progression free survival 4.5 years. And so again, the median overall survival not reached. And so a 5 year estimated of 75%. So we think increasing that follicular lymphoma should be really not ignored when it comes to thinking about CAR T. Autoimmune disease next year. We talked about autoimmune before. And this is saying that I just want to point out, I mentioned this once before, just last week or 2 weeks ago now in New England Journal, a study from China has been presented where they looked at CAR T for refractory autoimmune hemolytic anemia. For hematologists, this can be a terrible disease. And they showed basically that everybody, responded to CAR T. So, end of study, end of presentation. And like I said, I threw those together at the last minute, looking at some lessons from ASH. So we're telling that what are the lessons to summarize? Young donors are probably better than older donors. PT-Psi is going to be better than calcineurin inhibitor. 7 of 8 match and 8 of 8 match are now relatively equivalent. We're going to see graft engineered products that are going to compete with PTSI for outcomes, but that may be subject to being overpriced. And then we have ongoing improvements in the CAR T world where we're seeing new products for myeloma, including, for me, the most exciting first in human in vivo CAR T with proof of principle showing, damn, it's working. Um, that we're now seeing potential new endpoints to study in CAR T, which is going to be toxicity free, relapse free survival. We are actually seeing improved management schedules for advanced ICANS, and that follicular lymphoma, confirmed by 5-year data at the ASH meeting, is probably going to be a superior than, the various bispecifics or immune conjugates that people are offering. So I'll stop there. I still have, yeah, I did that 50 or almost 60 slides in 45 minutes. So that was my best I could do for you.
Great.
I hope that's useful, and I hope some of that's educational. I, for one, was excited by this year's ASH.